Thank you, Jen, and thank you for taking allowing me to speak in your curator slot. I'm Terry Meehan. I'm based at Embley BI, which is near Cambridge, England. As you can tell, probably in the and I really appreciate being able to talk to you because just a quick thumbnail sketch of myself. I did a PhD and a postdoc in mouse models of autoimmune diseases, but then curated at a large database at Jackson Labs for 4 years. And that was my first foray into informatics. And really appreciate all the hard work you guys do, and I'm happy to help contribute to your curation efforts. So I'm just trying to change my slide. Trying to move my slide forward. There we go. So why are mouse models of interest to curators looking at variants in human patients and people? Well, so rodents have been used as models for over 100 years to study abnormalities in human originally nutrition and then cancer. They're mammals with a 2-year lifespan, so this fits well in experimental setups for PhDs, postdocs, and so forth. There are well-established inbred strains of mice, meaning that the mice that are used, including with the IMPC, are homozygote for all their alleles because they're maintained through sibling breeding, and this allows for precise genomic precise functional analysis of targeted genes. There's many biochemical similarities between mice and human, despite the size differences. In fact, the first use of rodent models was in the discovery of vitamin A. And of course, there are differences, but these differences are well studied and understood. The mouse genome was the second genome sequenced after the human genome, and it came, I think, within a year of the human genome. And so it has paralleled efforts at the genomic level, and the technology has evolved for using mice as a tool right alongside the human. There's about 20,000, I don't tell you guys, there's about 20,000 human protein coding genes, and there are 14 to 15,000 one-to-one orthologs in the mouse. So for the vast majority of genes, we can study at a very simple level, and then in some cases there's duplications and deletions, but there are ways to work with that too. And up until the CRISPR era, it was the only vertebrate model where we could do genomic engineering. And I'll talk a little bit more about that. So the curators with your group, I found this table in some of your literature. You do actually rank model organisms quite importantly in your scoring in your experimental evidence summary, and I was excited to see that. And so I'll help explain how we could contribute to your experimental evidence scoring. So it's a bit confusing because mice have been around as models for over 100 years. There's many resources dedicated to using mice. On the left, and can people see my arrow? Does it circle around?
Yeah, I can see that.
Okay, well, maybe the laser pointer. Yeah. So there are commercial providers of mice, large companies, international, that maintain popular strains and sell these to researchers. There's a number of academic networks and resources that will deposit mice that researchers make and make those available to other communities. The MMRRC is an NIH-sponsored network, which all IMPC mice go to. And InfraFrontier is a similar network in Europe. And you can see there's many different national sources. This is just a sampling of what's out there. And there's a global catalog that tracks all this. The big elephant in the room for mouse resources is the Jackson Laboratory, where I mentioned I worked for 4 years. Based in Maine, there's also institutes in Sacramento and Connecticut. And it's an interesting organization. It's a kind of a two-armed organization. There's a business side that maintains and sells mouse strains. It's a nonprofit, so the money goes back into research. And then there's a research side, just like a university, with graduate students and applying for grants and so forth. And in that academic side is the Mouse Genome Informatics resource, MGI. MGI is a mouse genetic database that's been around for over 25 years. They do all things related to mouse genomics. They help do the gene annotations. They maintain a very strict nomenclature on the different strains and alleles and so forth. They bring in data around gene expression that is not RNA-seq based, so things like reporter genes and mutant mice. And one of the big things they did, and this is what I helped out with, was they curate the literature. They actually scan through all the papers and look for mentions of mutant mice, and then will annotate those papers and make gene phenotype associations. So to do all this work, they've been involved in a lot of development of semantic standards or ontologies. So MGI, along with EBI here and a few other places, were the founding members of the gene ontology, which probably many of you know is kind of one of the cornerstones of bioinformatics now. If you want to know what a gene's function is, the gene ontology is the first place you go. And they developed other ontologies around anatomy and mammalian phenotype, which I'll come to in a bit. So it's a complex ecosystem has evolved around model organism. And it's a busy slide. I apologize for that, but I'll walk you through it. So in the traditional realm of peer-reviewed science, most data is published in the scientific literature, as you guys know better than I. And of course, there's other resources. There's often supplementary data, bespoke databases for projects. And so over the last two decades, there's been these dedicated, what we call model organism databases that have been funded by the NIH, I think in large part by the NHGRI, to capture all this data. And so there's from yeast, worm, Drosophila, zebrafish, rat, MGI. They've all been working for I think all of them for decades to pull in data, largely from literature, in some cases from resources like this. As I mentioned, the gene ontology was in part an effort to collaborate across these resources. And this ended up being an important thing. I don't want to go into too many details, but all these resources now are working together on developing kind of an uber-mod known as the Alliance of Genome Resources. And so you may hear about that in the future. The NHGRI is facilitating that. But what's happened is by developing standards like the gene ontology and gene annotation and so forth, is they really provide a lot of resources to a number of tools and resources. So here we have, there's a repository, as I mentioned the mice, but all these model organisms have repositories where researchers can go to get the needed strains for themselves, to prevent duplication of efforts. There's large aggregators of data or resources that maintain genomic standards. And so I have these arrows pointing one way, but they really go back and forth. There's term enrichment, which is a very popular bioinformatic tool. If you have a list of genes, this is one of the first things you'll do is you'll do enrichment over that to see which Go functions are, gene functions are enriched in your list. And then increasingly, we're having some tools evolve around the phenotype standards, and I'll touch upon some of these as going forward. So where does my project fit into this? I already broke the first rule. I'm supposed to talk about my project right up front. So this is now I'm going to the IMPC. So the IMPC is a global research infrastructure involving these institutes you see here, with the NIH Common Fund funding the North American efforts and us in the UK. And this has actually been recognized by the G7 as a global research infrastructure, and we're trying to create and characterize a knockout mouse strain for every protein coding gene. For 20,000 genes. We want to publish and annotate this data in near real time, and this is where it is relevant for the curators. We just, as of last week, started year 9 of the project. It takes a long time to make mice and phenotype them, but we've got over 6,000 genes with phenotype data. And I'll come back to that. As we're not doing genes you really know about. It's genes that are that there isn't a lot of data for. These are the ones we selected. We do data releases 3 to 4 times a year, and as I mentioned, we're funded by the KOMP Common Fund. There's also many other national funding from throughout the world that supports this. So one of the things the Common Fund is supporting is our data research infrastructure, which is a collaboration of my team at EBI. I have 10 people working on this. MRC Harwell, which is an institute near Oxford, which is a mouse production facility and has experience with the mice. And my colleague Damien Smedley at Queen Mary of London, who is also part of a Monarch initiative, and where he's embedded in the clinical genetics that's happening in the UK health system, and so he's an expert in mapping phenotypes across model organisms to inform variant prioritization in clinical sequencing efforts. So the data flow is data will come from the mouse production centers, and it will flow to our data coordination center that's at Harwell. They'll check our data against standardized protocols. We'll check the data against our mouse tracking to make we track the mice here to make and help coordinate production efforts. And when data has been approved by the Wranglers to be of high quality, it's passed to my team, where we have we store the data, we analyze it, we match it to diseases, and we push it out. So I'll go into a little more description about those. So first, the alleles we're working with. So keep in mind, this is genomic engineering. We can do fancy things with these alleles. So before CRISPR came along, there was a U.S.-led effort to generate a bank of embryonic stem cell clones with targeted deletions of genes. And I won't go into all the details, but there were 2 technologies. One took out the whole gene. The second would take out critical exons and allow a deletion of the exon when bred to a certain transgenic strain. So we have some data from these mice in our first phase, about 3 to 4,000 of our knockouts are from one of these 2 technologies. But then CRISPR hit, starting about 4 years ago, and of course it's so much faster, it's so much more efficient and cheaper, and so now all the mice are having simple deletions of critical exons with CRISPR. So the strains that are selected to be made are done by the individual centers because they know their communities the best, and they know which mice are already available to the research community. We've all been working with genetic disease consortia like the Center for Mendelian Genomics, Kids First is now coming along, 100,000 Genomes Project in the UK, and so we've been making mice with null alleles, mice with alleles where the gene function is wiped out, to help inform these disease efforts. And our role is to help coordinate these efforts, to take in input from the centers, to make sure that there is minimal overlap. And it's actually been a very, very collaborative process. And if 2 centers want to make the same mice, usually they'll just quickly agree in an email, okay, you make it, and then send me some breeders as soon as you have them. So when the mice are after the mice are produced, we try and breed them to homozygosity, meaning that both copies of the gene have the mutant allele. And then starting at 9 weeks of age, when mice are sexually mature, they're put through a series of phenotype tests to assess different physiological systems. These tests are with the exception of a glucose tolerance test, they're not challenges. So one limitation is we can't infect the animals or do things that will impact subsequent tests. At 16 weeks of age, we sacrifice the mice, and the centers do an extensive necropsy to get as much data out of these mice. We have each one of these boxes has a community agreed upon standard phenotyping protocol, and each of these protocols has data parameters that are then in turn associated with ontology terms. I'll talk a little more about that. So one thing that was a little surprising when we first came into the project was the number of embryonic lethals we would have in these mice. So it ends up 1/3 of our knockouts are embryonic lethal, meaning when both copies of the gene are turned off, we do not get a mouse that survives to birth. And so the NIH supported a secondary, a large supplement to start looking at embryo morphology. And so this is where when a mouse is shown not to be viable through development, we go back and we look at embryonic stages. So a mouse a mouse embryo develops over 3 weeks, 21 days, and so we look at a midway point, what we call embryonic day 12.5. So that's 12.5 days after conception, and we do some gross morphology assessment. And then based on what they see, the centers will then go back and do an extensive imaging protocol earlier or later in development. And so this data is a little slower to come, but it's very heavy image based, and we do have some of this data available. And we're increasingly, the more and more we work with folks like you, we're realizing this data is actually very relevant to the human rare disease consortia, because oftentimes what you'll have is a hypermorph in a human, and we can see some very extensive annotations of this morphology, say around brain development or heart and so forth. And oftentimes you can these can be related to what you see in a human patient who has a hypermorph. We're also in the second phase of our funding. We're doing an aging pipeline. So we're taking several hundred of our strains and we're aging them for a year. So as I mentioned, we sacrifice the mice at 16 weeks of age in our standard pipeline, and that's the equivalent of looking at an individual who's 20, 25 years old. And so we did cost assessments and looked at what it would take to do a proper aging pipeline. The aging community would like to take mice out to 2 years, but that's just not feasible in a high throughput pipeline. So we're going to do a pipeline where we look at mice that are around the human equivalent of 40 to 50 years old, and we think we're starting that data is starting to come through, and they do have many more phenotypes. All data is, as I mentioned, is passed through the data wranglers at MRC Harwell, and we have a data wrangling platform we call PhenoDCC, where data is shared between the Data Coordination Center and the individual institutes providing data, and it allows wranglers to mark up abnormal data. Like here's a female control, and this is a mouse that has a weight that is much too high, and it's probably a decimal point shift. And so we can highlight that. The centers can look at that and say, yes, that was a mistake, and correct it, and we keep records on that. So it's very high quality data. When the data is passed to my team, we do an extensive statistical analysis on this. And so this is actually starting to get a little data. We have many more tests we're applying to the models as they come through. So we as data comes through, we look at the things like is the data categorical, continuous, image-based, so forth, and we put the best statistical test that we think appropriate to that, and then generate phenotype associations. One of the critical things we do is for all the continuous data, we use a linear mixed model, and so we model in body weight, sex interactions. So I'll mention this a little later. There is a lot of sexual dimorphism in our controls. We see sexual dimorphism in up to 40% of our parameters. And with genotypes and with the knockouts, we see sex-specific effects. With the knockouts, I think I'll come to that. It's about 12% of our hits. The sex will modifies what you see. There's also batch day-to-day variation, and we model that in. And because the mice are not tested all on the same day, they're usually tested over several days or weeks. And we can use that those different days to figure out the day-to-day variation and help make more robust phenotype associations. We use a p-value of 10 to the minus 4 as a way to control for false positives. This is kind of an empirical threshold, and this is one of the reasons we want to talk to you, because sometimes that's too conservative, and we don't want you to miss experimental evidence that might be relevant to you just because it's right around that threshold. So basically, I just threw a lot at you, and the basic thing is to keep in mind is that we make mice, we have standardized protocols. Each one of these protocols has these data parameters. So this is clinical blood chemistry, just like you would get if you were taking a blood test at a lab. We take the data, we always analyze by genotype, so this is homozygote, and by gender, by sex. We do our statistical analysis, and then we associate this with the ontology terms. So this would be increased circulating cholesterol level. So we're using the Mammalian phenotype ontology for these terms, and this is from our friends at MGI. And so here's an example where you're looking at abnormal pancreatic beta cell mass. And these, if you're not familiar with the ontologies, they're what's called DAG structures. And this is they're graphs where you have a parent-children terms. A child is always a more granular description of the parent, and in some cases you can have more than one parent. And we'll come back to the structure of these graphs in a little bit. So overall with the project, we're making mouse models 60% of the time for genes that have never had a mouse model before. And where we do have overlap, it's because the mice are only described once or twice in the literature, or they're hard to get, and so they want to make these mice available to researchers. And over half the time, almost half the time, we're adding new experimental evidence for gene function where none's existed before. So gene ontology has these annotation codes where they make an inference to a function that can be experimental, curated, automatic, electronic. So many times you'll see something like binds a metal ion, and that's just an automated electronic annotation that doesn't really tell you a lot. And even in cases where there is experimental evidence that's been there previously, as you guys probably know, when you dig into the primary literature, you're usually very focused on one pathway, one biological system. And what we're contributing is a very broad overview. So we're complementing a lot of the research that's published in the literature. So I'm going to shift over to the website a little bit because this is our primary way to get data, but not the only way, so I'll explain. We just launched a new version of our website in May, at end of May, and it's interesting. It was an exercise of removing stuff. We had too much on there. This is based on UX testing, and so it's kind of a streamlined, faster a website to really get people to the gene phenotype data, because that's what people really wanted. So we focus on searching on genes and phenotypes. I'm just really going to focus on looking at how we look at genes today. And so you can search for a gene, and you can use a mouse or a human gene symbol. We have all the mappings to the human gene orthologs. You can use a gene name or MGI ID. And so in this case, I'm looking at a CYP17A1. When you hit return, you'll get a quick summary of what we have. So the gene name, the orthologs, any synonyms. Then we also have some mouse-specific information. Is there a mouse produced? Is there phenotype data produced? So when you click on that, you'll go to a fairly long gene page with modules. And so the modules are arranged for gene information, phenotype associations that we have, images, disease models, and where you can get the mice. So to walk through those different modules. So the gene information, this is, we used to show a lot more and we were losing people. So we stripped it down to some of the bare minimum, some of the ideas I just mentioned, whether the mice are viable or not, links to MGI and Ensemble. And we give a phenotype overview, and this is something that UX has tested very positively on. So each one of these represents a physiological system. And if it's blue, it means it's been tested and we didn't find anything significant. If it's in orange, it means we found a significant phenotype. And if it's gray, it means we didn't get phenotype data for that yet. We also have a body weight curve, as that's important for mouse biologists. We have icons to other modules, and I'll come back to those. So we're going to look first at the significant phenotypes. So if you click on this, or our default is right below this gene summary, is you'll have a phenotype table. So this particular knockout has many phenotypes, and so this is in a table that you can select. You can sort by the column. In this case, I've sorted for smallest p-value, most significant p-values. We have the MP phenotype term names. We have the system they're associated with. We have the alleles, and this is specialized nomenclature describing the allele that was generated. I won't go into it. I could go on and on because I was a curator in MGI. There's zygosity, so this means it's homozygote. And we also will test heterozygote adults if the embryo homozygotes are lethal. So if we get enough het animals, they'll be tested. And then whether the phenotype is seen in male or female or both. P-value and life stage, this is for the aging. As we get aging data in, we'll start putting in weeks of age. So if you click on any one of these, you'll pull up a data page. So the data page here describes the number of mice we've tested, and the protocol, and a box plot of the data, and a timeline of the data. So one thing is very important about our resource that we have trouble getting across is that we've been doing this for 8 years, and we test a lot of mice, and every week control mice are tested. So we've built up this incredible baseline. So for this particular experiment, and this is one of our partners from China who came in later into the production, we have data from 844 control mice. And for the mutants, we try and look at a minimum of 7 males and 7 females for most of the adult tests. In this case, they're 8 and 7, 8 females and 13 males. So it's a large amount of data, and we're actually now starting to window some of this data. We always track the metadata. If the metadata changes, we would create a split. But even over time, is data collected in January of 2014 relevant to something a year later, 2 years later? So we're actually working on a pipeline, and we have a paper that's near about to be accepted in Bioinformatics that would just select the best window of control data to cut down some of that. So we're always trying to improve the stats, and that's the next slide. Is if you like stats, below each one of these graphs we have many stats that are generated by our analysis pipeline, but the most important being the p-value for the genotype and the classification. Do we see any sexual dimorphism in this? And as I mentioned, we do see sexual dimorphism quite readily. I'm sorry, that's a bit blurry. This is an example where for cholesterol, we see no difference between wild type and homozygote knockouts, but in the males we see a significant difference. And as I said, with our knockouts, almost 13% of the time we'll see a phenotype in one sex and not see it in the other. 3.5% of the time we'll see different directions, meaning we'll see high cholesterol in the males and low cholesterol in the females. And so what's that, about close to 80% of the time the observations are equal across the sexes. So we have a paper in this. It's one of our more cited papers in sexual dimorphism. It's a very real thing, and of course as biologists I think people are appreciating that more and more. So going back to the gene page, I showed you the significant phenotypes, but something that is relevant to curators is you may want to look at all the measurements. And so we have this all measurements icon here, which we also have a tab here. So if you click on that, you'll see all our continuous data measurements that we have. So this is a work in progress. The UX people said the biologists really like this, and so we're paying more attention to it. The problem is we have so much data that we can't fit in all the rows, all the labels here in the columns. So what this is showing is p-values, a minus log 10 p-value, where anything above this green line, anything to the right of the green line is considered significant. So currently, if you want to use this, the best way to look at it is to hit this deselect all button. So we have this deselect all. So if you hit that button, it removes all the data, and then you can select your tests of interest. These are different procedures. So here I've clicked on clinical chemistry, and then you can look at the data in a sensible way. So here's the clinical chemistry, and when you find a data point you like, you can click on it, and it'll give you some metadata, and then click on it again, and it will give you the chart page. So this is something that users really like, and I think could be of interest to you. In this case, if you're looking at CYP 17A1, if you're looking at a human variant, and they were reported to have high albumin, you can see here albumin was just below the threshold. But you could click on that, you could look at the data, and it could provide experimental evidence. And this is the type of thing that people who are taking our data summaries will miss. And so this is where we think going to the portal or getting a report from us would be beneficial. Then I want to talk a little about disease models. I think it's important for this group because we're doing something pretty neat and with our collaborators in this space. So I mentioned the MP ontology that's used for annotating the mouse models, and in this case, it's not we're doing this for IMPC mice, but MGI uses the same ontology for curating mice in the literature. Now, there's the human phenotype ontology, which you may or may not be aware of, but if not, this was developed by Robinson, originally at Charité in Berlin. He's now at Jackson. And what he started off with, he went through OMA and he started looking at all the traits that people were describing in the free text and started developing an ontology around that. And he knew the MP existed, and he liked the structure of it, so he modeled it on that. But he used clinical terms, as he should. And of course now it's taken on, it's a very popular resource, and you guys are probably aware of it. My colleague, a fellow IMPC PI, Damien Smedley at Queen Mary University, as I mentioned once, he's part of a Monarch initiative with Peter Robinson, Melissa Handel at Oregon State University, and others. And they've been generating and refining and using graph mappings across the different phenotype ontologies to help prioritize variants by the phenotypes that are visible. So if you have a human disease population that has these clinical observed phenotypes, these ontologies can be used to look at similar clinical terms with another patient profile, but also with orthologous phenotypes observed in model organisms. So if we look, and the key is that it's not a lexical matching. It's built on these graphs. And we can go up the graph and can traverse up and down the graph. So we can link something like optic nerve swelling in a mouse model to optic atrophy in a patient profile, or optic nerve hypoplasia that might be observed in a new patient coming into a resource. And there's a lot of there's several publications in the space if you're interested in the math on that. So what we do is we push all our phenotypes to Damien and the Monarch Initiative. And ask them to look at our how our models match up with human disease populations. So if you go to a gene and you hit the disease model button, you'll jump down to this little table. And so what's happening with this table is we have two tabs, and we default on by annotation. What this means is that we're looking at genes annotated by Omen or Orphanet or the DDD program done by the Sanger Wellcome Trust, and we say this mouse gene is orthologous to this human gene that's been associated with these three diseases, 2 by Orphanet, 1 by Omen, and then we look at our phenotype scores, and you can look at this last one. This is our phenotype, and we look at the percentage of phenotype overlap, and so 100% is perfect and 0 is bad. And we think anything above 50 is worth looking at. When you can click on this row and it will bring up the different models. This is the one area where we bring in outside phenotypes as curated by MGI. So we have an MGI strains here that have been curated from the literature, and we have our IMPC strain with our phenotype data. And if you below this little table is this widget provided by Monarch that looks at the phenotype overlap between the mouse models and the human clinical terms. And this is our IMPC mouse model. So you can see that we've picked up on 1, 2, 3, 4, 5 terms we overlap with. And because we're using graphs, they're not perfect lexical matches, but we can, the graph moves up the graph up and down a little bit. So for us, we wouldn't annotate something called with osteoporosis. We would annotate to decreased mineralization of bone. And so this is proven to be really one of our killer apps because we're doing genes that are not well characterized in the literature. And so this is feeding forward to folks like you. So the second thing we do is that we can ignore the gene orthology. So that example I just showed are diseases, human diseases associated with human CYP17A1. Instead, now if we ignore the genes and we just look at phenotype overlap, we can match up to other disease patients. In this case, we're hitting a lot on spermatogenetic failure because CYP17A1 mice the males are infertile. So we're pushing this idea of phenotype orthologs. And my colleague Damien with Monarch has developed a tool called Exomizer, which is taking the typical route to find rare disease variants in human disease populations. So taking the whole exome, removing off-target and common variants, and then taking the variants that are left over, and then scoring them by how the variants, the genes are in, what are the phenotypic profiles of model organisms, and then coming back with a prioritized list of variants from this patient that otherwise had several good variants that could be considered. So this Exomizer tool started off as kind of spun off from this project with Monarch several years ago, and Damien got involved in the NHS genomic medicine. So NHS is the socialized medicine we have here in the UK. And it's now being incorporated into the genomic medicine pipeline at NHS. And he got it ISO certified, so it's suitable for the clinic, and it recently got picked up by a commercial company here called Kagenica, which is in the human genomic space, and they're promoting it as a good way to prioritize variants. But it's still freely available if you want to run it yourself, and I have the GitHub there. So just a couple other things before I finish up is we have data reports that we provide to different collaborators, simple SCVs. We would be happy to build a custom report for the curators in ClinGen if there's particular things you want. And as I mentioned at the beginning, the mice are they're deposited in repositories and they're being used. It's still cheaper to buy a mouse from a repository than make one in this era of CRISPR. And we have over 2,500 papers we know of that are using our mouse strains and increasingly just using our phenotype data, and that's included in here. And so so it's nice coming back to that ecosystem. We're getting data out through the website, as I've described. Sorry, that's the old website. Should I update that slide? We have these data reports. We have APIs too. If you have an informatics group that wants to take in our data and bring it into your resource in a way that's helpful to you. And we're pushing the data out. And so the data is being used by the Monarch Initiative, and they have a nice portal to look at phenotypes across model organisms and disease populations. Our data is going right in MGI, so MGI has our phenotype summaries. They don't have the underlying data, but MGI is already piped into a lot of different resources. So we made that as one of our top priorities, and the data is getting into other places. And the nice thing is starting to infer the ecosystem is, as our mice are being used by other researchers and they publish in publications, then MGI annotates those publications. And so this is perpetuating and helping the community. So we just started year 9 of funding. We have 2 years of funding left. We're hoping to get another 5-year chunk that we believe will allow us to finish all the one-to-one orthologs, the 14,000 to 16,000 one-to-one orthologs. But we're also starting to push the state of art in genomic engineering, and most of our partners are are now investigating best ways to get point mutations, deletions, more complex alleles that directly model the alleles that people are seeing in human clinical samples. And there's also there's some conserved non-coding elements. Non-coding elements are not nearly as well conserved as protein coding elements across species, but there are conserved ones, and we're starting to investigate those. So we have a 10-year plan. So we have a plan to 2030, and so hopefully we can keep contributing to resources like yours. So in summary, the IMPC is generating new broad-based gene phenotype associations. It's unbiased analysis and phenotype calls. The data is really robust. We can confirm what's seen in other publications, and oftentimes we'll be adding new knowledge. We provide this phenotype overlap scoring to disease models and an ability to explore the data, so you can dig in and see if there's things that didn't quite make our phenotype call, but still could be significant. And we're using community standards so that the data can get out to other resources. And I'll finish on my summary slide. There's a lot of different funding bodies that help us, but the biggest by far is the NIH Common Fund, which is KOMP. And so I used IMPC today, but we often call ourselves the KOMP project too, because their contribution is so large. And with that, I have a few minutes for questions.
Great, thank you. Yeah, I think as you showed earlier in your slides with the ClinGen matrix, having an animal model can really influence our clinical validity classifications. So this is a really useful tool for our curation process. So we appreciate you taking the time to talk to us today. So as Dr. Meehan said, we have just a few minutes before we have to sign off. Does anyone have any questions?
No other questions from here.
I'm trying to remember if you went over on your slides, Dr. Meehan, if you have posted or can provide in a link a PMID to the International Mouse Phenotyping Consortium or Knockout Mouse. That's one of the things for us to actually score this information. We need a PMID for it.
Sure. Yeah, our landmark paper was in Nature, and I can provide that.
Thank you.
Yeah.
I have a question about prioritizing which genes to knock out. Is there any mechanism for someone in the community, for instance, to provide feedback on, let's say they have a gene-disease relationship that, there's quite a bit of evidence out there, but maybe a mouse model would really show a definitive association. Is there any mechanism to provide feedback on a gene that they might want a knockout for?
We're trying to formalize that. We have a web email you can contact us, and we do monitor that quite closely. And we have these collaborations set up with CMG for one, and Kids First is second, where the PIs talk to each other occasionally. We're actually going to have there's going to be a satellite meeting at ASHG in this space, and if anyone's interested in going, we're looking for some people doing the type of work you're doing to find out how we can better that communication on the portal. So I'd be happy to. We're going to reach out to some of the PIs in ClinGen in the next week or two about the satellite. But right now it's mostly the centers talking to the PIs of the various resources. So Baylor is oftentimes taking requests from CMG or UDN and so forth. And this is an area we want to target more.
Okay, yeah, that's great. That would definitely be a lot of interest to ClinGen. Just thinking about how many moderate gene disease associations we have, that having one mouse model would probably make them definitive. So that would be great.